The purpose of this experiment was to calculate the concentration in parts per million of chlorine that is present in the fountain water at GHS. We collected samples from the Science Building, the Math Hall, the Fine Arts Building, and the Language Hall. We made sure that the water bottles we collected the water in had been washed and dried the night before so contamination could be minimized and controlled. Before we could begin, we had to make two solutions that would be required in the experiment. First, we made a starch solution that would act as an indicator. This was done by placing a beaker of about 25 milliliters of water onto a hot plate and heating it up until it boiled. Next, we added in a sufficient amount of cornstarch and stirred until it was all dissolved. The amount of cornstarch or water put in did not really matter because it was only acting as an indicator. After it was dissolved, we turned off the hot plate and carefully removed the beaker and let the solution cool down. Our solution ended up being milky white in color and a little thick. The next solution required was a 0.025 molar solution of sodium thiosulfate. To make this, we measured 25 milliliters of water in a graduated cylinder and added 0.0988 grams of solid sodium thiosulfate into the graduated cylinder. Using a stirring rod, we stirred the solution until it was completely dissolved. In order to help speed up the process and ensure that the particulate was dissolved, we cupped the top of the cylinder and shook it for a while. With a pipette full of the solution, we counted the number of drops it took for the solution to reach 1 milliliter in a separate 10 milliliter capacity graduated cylinder. We repeated this 3 times so that we could measure the average volume of solution in 1 drop. After the solutions were made, the experiment could begin. Starting with the Science Building, we filled a beaker, which was cleaned very thoroughly beforehand, up to 300 milliliters. We left the other samples in their bottles to prevent unnecessarily and prolonged exposure to the air. The first thing we did was add 1 gram of potassium iodide into the beaker. Because chlorine is a better oxidizing agent than iodine, the chlorine from the water displaced the iodide, which was visibly evident in the yellowish color from the free iodine. Next, using the pipette, we added 3 drops of the starch solution that was made earlier into the large beaker, which caused the yellowish solution to turn into a dark blue color. This was caused by the contact made between starch and iodine. Taking 50 milliliters from the large beaker with a graduated cylinder and pouring it into a smaller beaker, we slowly added drops of the sodium thiosulfate solution using a pipette until the color of the overall solution turned from dark blue to colorless. While one person was adding and counting the drops of sodium thiosulfate, another person made sure to keep swirling the water around. A white paper was placed under the small beaker with 50 ml of water solution so that the person adding the drops could see the change in color better. Once the titration was complete, the contents were poured out and the beaker was cleaned very thoroughly. This was repeated 4 more times using a sample of 50 ml of water each time. Once the 5 trials were complete for the Science Building, we repeated the whole process with the beakers of water from the Fine Arts Building, the Math Hall, and the Language Hall. The number of sodium thiosulfate drops used to titrate the water in potassium iodide solution were recorded and can be seen in this table. After several calculations, which can be found in the formal write-up of this experiment, the average concentration of chlorine from each building was calculated, which can be seen in this table.